Hello, welcome back. So we're going to continue learning about different imaging modalities in the context of radiotherapy. The next one on the list is PET imaging, positron emission tomography. And this, by the way, is easier to understand, I think, than MRI. So it's not quite as intense. So I'm going to go over the basics and try to hit on some of the applications in this as well. Here we go. Before I talk about PET specifically, let me back up and look at radioisotopes in general in nuclear medicine. So here's a list of some of the radioisotopes that are in common use, just a selection of them. And you'll see that they're produced by various processes, they undergo various decays, and they have different half-lives. So to understand this, you're probably going to want to go review the decay modes that we learned about in video 2.2. We learned about here beta minus decay as one of the decay modes, and that produces certain particles, electrons, and also photons come out. Also, you'll remember beta plus decay. That produces the positron, the beta plus particle, the opposite of the electron. And then that electron goes out in tissue and does things, and we'll look at that in a minute. So those were the two decay modes, and you'll remember that you had the neutron-rich isotopes and the proton-rich isotopes, and they decayed by two different processes. So before I talk about PET, let me talk first about SPECT. That's single photon emission computed tomography. And that works on these radioisotopes that emit photons, and you can image the photons. So the geometry is this. You have the patient with the isotope that has accumulated in the tumor here. That isotope decays and emits photons, and it goes up into the system. So what you have here is a scintillator, okay, that turns particles into optical photons. And then behind that, a position sensing photomultiplier array, so it can tell where the particle landed on the face of this detector. This is a two-dimensional planar detector. And then in front of that, you have a collimator. Okay, and that means that particles coming in this way, for example, can get through, but coming in sideways, they can't. And so that gives you kind of an imaging, a low-resolution imaging capability. Now, this whole assembly will rotate around the patient and acquire images. It rotates slowly, acquires images at different angles, and similar to what we saw with CT, you can do tomography, back project, and figure out what the distribution of isotope in the patient is. Now here it's an emission tomography, so it's photons coming out of the patient instead of being transmitted through, but the mathematics are similar in many ways. Now let's talk about PET, positron emission tomography. PET relies on an isotope that undergoes beta plus decay. And I'm showing the classic one here, which is F18, fluorine 18. Here's the decay scheme for fluorine 18. You see that the half-life is about 110 minutes. It decays in two different ways, but mostly through beta plus decay, which is the one that we're interested in. And the beta plus decay produces a positron, and that positron is used for imaging, as I'll show in a minute. Now, of course, to do something for oncologic imaging, you can't just inject raw F18 into the patient. It has to be conjugated to a molecule that does something of interest. And so let me go through the biochemistry of that for a minute. So we're talking here about F18 fluorodeoxyglucose. So that's a glucose analog that's taken up by tumor cells, these cells that are in anabolic metabolism. And that's an altered cell metabolism in cancer cells that's been known about since the 1930s. And like glucose, this compound is taken up in the cell. That's done actively through glucose transporter proteins. Then it's phosphorylated and goes through this glycolysis process. However, the FDG can't undergo glycolysis, and that's because the OH group has been replaced, so it's not metabolized. So bottom line, it's trapped in the cell. And so that's what allows you to image the location of the tumor cell. So how that works in practice then for imaging is the patient will come in, they'll fast for at least 6 hours before the scan to get the glucose levels down. Glucose levels should be low at that point, they're checked. Then there's an injection, an IV injection of somewhere between 50 and 20 millicuries. And the patient waits an hour, and there's no activity. That's to avoid uptake in the muscles. And then the imaging begins. So now let me go through how the images are acquired. So you have a patient here who's going to be scanned, and the injection happens, and the FDG accumulates, and let's say it's accumulating into this tumor region here. Zooming in then, what I would see is an F18 molecules here. They would decay then into the 18 oxygen to that beta plus decay, and then create a positron. That positron has some energy, and it wanders around in tissue until it slows down, and it comes next to an electron here, and you undergo electron positron annihilation. That creates 2 photons. The energy comes out as photons. And so you get 1 photon going down, let's say in this direction. The direction can be kind of random, but let's say it goes down this way. And then because the momentum is conserved, you have to have another photon going out the other direction so that there's no momentum before and there's no momentum afterwards. The 2 momentum vectors cancel each other out, right? So to zoom out then, you have the tumor being imaged, the 2 photons coming out, and then around the patient, you have the PET detectors. So this is a ring all the way around, and it's segmented into blocks, and inside each of these blocks are scintillating crystals, so that when a photon interacts with the block, you can read that out. So the energy of these 2 photons coming out is 511 keV in one and 511 keV in the other. And you know that because of the energy conservation. So you started out with the positron that had 511 keV, an electron that had 511 keV. So afterwards you have to have the same amount of energy. So that's where that comes from. And this is important because now the detector can be tuned to accept only photons around that energy. And so that allows it to reject all the photons that are outside that band, like for example, if there's some scatter events or so that go to a lower energy, you can reject those. And so because of this, the sensitivity of PET detectors can be extremely high. So these photons go out and they interact in the crystals in the PET detector, one on each side of the ring. And so you get these coincident events that happen almost at exactly the same time, one and the other. And because of that coincident event, you know that they were created somewhere along this line that connects the 2. You don't know where, but somewhere along this. It's called the line of response. Okay, so that allows you to conclude it's somewhere along this line. And then this is the tomography part of this. So like CT, you're taking different projections, right? Coming at when different events come, they give you different lines of response, and you can deconvolve that and reconstruct and figure out what the underlying distribution looked like. So one thing worth noting is that these photons don't come out exactly opposite to each other. There's a little bit of an angle you can see here. And that's because there's a balancing momentum to the nucleus that they came from. So it gets kicked in the opposite directions. The photons have to move away from that a little bit to balance the momentum. And so the line of response then doesn't run exactly through where the annihilation event happened. And that's one of the things that affects resolution in PET images. There are other things. So non-collinearity of annihilation photons, we just talked about. Crystal size in the detector. The energy of the emitted beta plus particles, and that's because they wander around in tissue before the annihilation happens. And that gives you a smearing because the point where the annihilation happens is not exactly the same as the point where the decay happened. And the higher the energy of the beta plus particle, the farther it will go, and the more that will affect the resolution. And then also issues with reconstruction, filters, and algorithms like we saw with CT scanning. So let's just have some look at some images. This is the CT, and then a PET is acquired at the same time. Here's the PET. You can see the uptake in the liver. Let's scroll through the CT, just have a look up in the lung. And this is a patient with lung cancer. I don't know if you saw the tumor there. Let's scroll back, find it. Okay, so there we go. This is what we think is tumor. Let's look at the PET scan now for this. So it's fused. You see the resolution of the PET scan is quite a bit worse than the CT, right? But you have this fusion that helps you visualize. So the tumor uptake there is well visualized in this patient. All right, now there's a lot of non-specific uptake, like you'll see activity in the heart there, the liver. As it's being cleared out, you'll see the kidney, activity in the kidneys. If I can scroll down and find the bladder, you'll see lots of activity down there as well. There we go. So good for staging and very sensitive. So a couple of things to say about this image. One, the PET resolution is not as good as CT, as I said. Two, these PET values are represented by numerical indices, which provide some indication of the tumor burden in here. And that's called the standard uptake value. And it's basically taking the injected activity normalized by the body mass. Here's another view of the same patient at coronal cut, and you'll see again the uptake of the FDG in the brain, the heart, the liver, bladder. The tumor is in another cut here that you can't see. Now on the right here, I'm showing what the image looks like if you don't have attenuation correction. And let me explain what that means. So if we go back to this picture and we imagine annihilation events happening here, you have photons coming out. Let's say some are coming out. Let's look at this direction, and then the opposite direction will be out here. Now if you notice here, photons in this direction, they're going through the lung. Because the lung is less dense, you actually get many more photons out this way because there's less attenuation here. If you don't correct for that, you're going to get some artifact because you're going to artificially think there's too much emission coming here, although it's really just an attenuation effect. So this is what we call attenuation correction. And if you don't do attenuation correction, then you get the image on the right here. So you'll see here that there's lots of activity in the lung because it's not attenuation corrected. You'll see extra activity around the surface, around the skin. You'll also see some kind of like bright enhancing artifacts near the edge of some organs, like the liver here. It looks like there's a big enhancement on the lateral edge. So you need to do attenuation correction to not be fooled by those artifacts. All right, now up to now, what we've talked about with PET is FDG PET, so imaging this fluorodeoxyglucose, but we should note that PET is not just FDG PET. You can use other compounds and other isotopes as well for other biological processes. So here are some examples that are used in oncology and also other studies outside of oncology. And so you can see a wide range of different tracer compounds. So, and also you'll see the isotopes are different: carbon-11, oxygen-15, F-18, in different ways. And so here's a list of the isotopes that are used, and you'll see they have quite different half-lives and energies of the positron and range in tissue for the positron. Interestingly, they're mostly made by cyclotrons. That's because they're proton heavy if they're going to decay by beta plus decay, or sometimes there's a generator for these. Some of the shorter half-life ones are quite challenging to work with. Oxygen-15 would be great to measure perfusion, but it's got a half-life of 2 minutes, very challenging to work with. Okay, there you go. Those are the basics of PET imaging. Hopefully this is helpful. Here's a list of further reading materials if you want to go study some more. And in the next video, we're going to come back and learn about another important imaging modality, ultrasound. Thank you.